All right, so we're talking about meiosis in this section. So we talked about mitosis in chapter 5. Meiosis we mentioned briefly yesterday. We'll talk more in detail about that process. So during meiosis, diploid cells undergo 2 cell divisions that result in haploid cells. So mitosis, we just see 1 division. You start with 1 cell, that 1 cell divides into 2 cells, and that's the end of mitosis. For meiosis, it's 2 of those cycles. 1 cell becomes 2 cells. Those 2 cells then divide again to become 4 cells at the end. That's what it's telling you in this section. I don't know why it keeps on showing that I'm talking right there. Usually it's up at the very top. Don't know how I can change that. Oh well, I guess we'll just keep it. Hopefully it'll go away. So as I mentioned, cells undergo 2 rounds of division and meiosis. I already showed this diagram yesterday, so don't worry about copying this down again because we already did copy this down. But just to go through it briefly again, meiosis makes 2 genetically unique cells. So mitosis makes cells that are the same. Meiosis, they're all different. It results in haploid cells, and this takes place only in certain times in the life cycle of an organism, only to make egg and sperm cells. So that's the job of meiosis. Meiosis reduces the chromosome number and creates genetic diversity. So what it does is it cuts the number in half of chromosomes. You can see in this picture, you start with 4 chromosomes. You have a green, red, purple, and blue chromosome. They divide into 2 different cells, and each of those cells only now has 2 chromosomes. So it went from 4 to 2 chromosomes in each cell. And that's what happens in our body cells too in meiosis, except that we had 46 to start with. It goes from 46 to 23 chromosomes in the egg and the sperm cells. So mitosis has the same number of chromosomes start at the end, but meiosis, it always halves the number of chromosomes from 46 to 23 for a human. Meiosis 1 and meiosis 2 are the 2 stages of meiosis, and each one has 4 phases, and they have the same names as in mitosis.
Can you please go back?
Sure. So meiosis, it's not hard to remember the stages because it's the same: prophase, metaphase, anaphase, telophase, just like we saw with mitosis. They look a little bit different, but it's the same names in the same order of steps. So this chart, again, we already have that in your notes, so don't worry about the chart, but just the bullet points, that's important on here. Okay, so meiosis 1 and 2, again, have 4 phases, same names as in mitosis. What happens in meiosis 1 is that you have homologous chromosomes that separate. So sister chromatids split apart, or sorry, they don't split apart, but they separate 2 different cells during meiosis 1. So homologous chromosomes are similar chromosomes, but not identical to each other. Well, I was hoping it would go away and you could see it. Try this instead. It's not going to help anything, is it? So in meiosis 1, sister chromatids stay intact. They still stay as X-shapes in meiosis 1, but the homologous chromosomes themselves are divided into 2 different cells. Then in meiosis 2, the sister chromatids split apart. And so that happens when the X's split. Homologous chromosomes, remember yesterday that, each one comes from your different parents. So see, the purple comes from your mom, green comes from the dad. They can have similar, but not always the same genes. Sorry. So sister chromatids are copies of the same exact chromosome, while homologous chromosomes have similar but not always the same genes. So sister chromatids are each of these, there's two right here. So there's two purples, they're identical copies of each other. And the purple is similar to the green. They carry the same kinds of genes. They might have the same genes for eye color, for example. But one might be for brown eyes, one might be for blue eyes. So they have different genes expressed, but they code for the same kinds of genes. But the green ones would contain, let's say this codes for green eyes. There would be two genes for green eyes, both on each of these sister chromatids. This one might code for brown eyes, and so there'd be two genes on here, each for brown eyes. And they're separated in meiosis II into individual egg and sperm cells. So here's the process of meiosis. So in interphase, you have condensed chromosomes. They duplicate in prophase. They become X-shaped. In meiosis I, the homologous chromosomes separate. So do you see right here that it still stays as X's? They don't have the sister chromatids split apart, but you do have one chromosome go into one cell, the other go into another cell. And so, they're split that way. And then in meiosis II, sister chromatids are split. And now we have single chromosomes in each of the cells. So it goes from diploid to haploid. This has two chromosomes. This is only one. Now you might wonder, well, why does this count as one when we have two parts to it? Well, they're the same exact genes. They're identical to each other. So we only count this as one. This is one chromosome, duplicated chromosome. This is one unduplicated chromosome right here. So these only count as one in both cases, duplicated, unduplicated. This counts as two separate chromosomes. They code for different genes. It goes from 2 to 1. Now this is simplified. Again, for humans, it's going to go from 46 right here to 23 chromosomes right here. Meiosis I happens after the DNA is replicated. So the first step before meiosis begins, just like in mitosis, is the DNA has to duplicate itself. And that's what we see as X shapes when that happens. And then meiosis I divides homologous chromosomes into 4 phases. And so we can see that there's prophase I, metaphase I, anaphase I, telophase I. So those are the 4 steps of meiosis I. So you start with chromosomes, they're duplicated, they line up in metaphase. During anaphase, though, the difference between this one and mitosis is that in mitosis, sister chromatids are split apart during anaphase. In this one, that's not what's happening. For this one, you have homologous chromosomes. Lucy, if you could try to restart your computer or restart Zoom, that usually helps. I don't know if you hear me or not, but I'll try to send this to you too. Zoom freezes all the time. Don't know why. And then you can see right here that there's still duplicated chromosomes. So they're still X-shaped at that point, but you went from having 4 in this cell to having 2 in each cell. Does everybody have those down? Top 2 bullet points? Okay. Meiosis II divides sister chromatids into 4 phases, as we talked about. So meiosis I divides homologous chromosomes. Meiosis II separates sister chromatids. That's the difference between the 2 stages. You can see right here that it's moving from duplicated sister chromatids to being single sister chromatids. So in both cases, you have 2 chromosomes to start with, but these are duplicated, unduplicated chromosomes. Sorry, let me go back. Didn't mean to do that. Okay. So DNA is not replicated in between meiosis 1 and 2. So the DNA stays the way it is. You just have two chromosomes, X-shaped chromosomes that are separated during anaphase. And at the end, in telophase 2, you now have 4 haploid cells. Each cell has a single strand of chromosomes, and we have 4 cells from that. So this is just an overview of what we talked about. You can see the two steps. It's a meiosis 1 on this side, meiosis 2 on that side. Yes. Oh, sorry. Here we go. That's what we're on. Everybody good? Okay, so meiosis 1, you can see right here. So we have homologous chromosomes. So we have 2 blues, 2 reds. Now, I didn't mention this, but it's important to understand what's happening in this step right here. Do you see how in metaphase, keeps on doing that, sorry. Do you see how in metaphase right here that they're crossing over right here? And then what happens down here? Do you see how it has some fragments of red on the blue and some fragments of blue on the red? Can you all see that? Yeah. So what's happening? Well, the genes are swapping. So during metaphase 1, they have what's called crossing over of genes, where the genes will swap places with each other. And at the end, what do you have? Well, you have red and green red and blue intermixed in these genes right here. Why is it important? Well, it's like shuffling a deck of cards where the genes are moved around, and it gives you more possibilities of traits in the offspring. And so what happens is that, now think about the baby being born. So where are these genes from? For the baby, that's from their mom's mom and their mom's dad. So that's their grandparents. So what's really happening here is you have the grandparents' genes, both on the mom's and the dad's side, being swapped around. And that gives you a greater possibility of traits in the children. So that's why it's important, because if you didn't have crossing over right here, you wouldn't have this wide variety of traits in the siblings that you see. So after crossing over, you can see right here that you have a mixture of genes. They're duplicated sister chromatids, they're X-shaped. And then meiosis 2, the sister chromatids are split apart into 4 cells. Okay, so there's a few differences, as I mentioned, between meiosis and mitosis. One difference, as we've talked about, is that in meiosis, we have 2 cell divisions. Well, in mitosis, you only have 1 cell division. In mitosis, homologous chromosomes never pair up. We never see the mixing of traits. You just see the division of sister chromatids. The last thing is that in meiosis, you get haploid cells. Mitosis, you get diploid cells. Does everybody have these down? Starting. Sorry, let me go back. Everybody good on this? The other thing that is different between the two is that in anaphase 1 of meiosis, sister chromatids remain together, but in anaphase of mitosis, they are pulled apart. Okay, so meiosis's purpose is to create egg and sperm cells. So let's go through it in detail what that looks like for sperm versus egg. The production of egg and sperm cells is called gametogenesis. Gametes is just a general word for egg and sperm cells. So gametogenesis differs between male and females because they produce different things. Oh, just a reminder about notebooks. I will take up your notes next test. That will be before Christmas break. So we have the last test of the quarter, most likely will be chapter 6. Well, maybe we'll see about chapter 7. But chapter 6 test, I'll take up your notes and grade them for your notebook grade for the quarter. If you miss a day, make sure you get notes from somebody and you put them in your notebook. So we can see on the right-hand side, this top picture shows you how sperm are produced, bottom picture shows you how eggs are produced. You can see that they look quite different in how it's done. For sperm, you can see they become really small. So sperm's purpose is really just to transfer DNA, and that's it. So that's all they really have is DNA and then the tail to swim. They don't have much else to them. So they're very, very small cells. So they're streamlined and they're motile, which means that they swim through water. That's what motile means. So as they move through this, from meiosis 1 to meiosis 2, they get smaller as they go through the process. So again, the main purpose of sperm is just to contribute DNA to the embryo, and that's really all they carry to the egg is just DNA from the father. If they were bigger, they wouldn't be able to swim very far, and they wouldn't be able to reach the egg. And so that's why they're so small. Eggs, though, are very big because they don't have just DNA. They need to have cytoplasm and all the organelles for the new baby, along with nutrients. And so the bottom picture shows you the cycle for making an egg. And what we can see right here is that this is meiosis, but you get 2 cells, but only one is usable. The other one is called a polar body. I mean, it just dies off. It can't really be used to make egg cells. Why does that happen? Well, the eggs have to be big, and so it's uneven division. The left-hand side gets a lot more of the cytoplasm and the nutrients and the organelles, so it can get large. The other one is too small to grow, and so it dies off. And then again, the same exact thing. This one cell right here divides into 2, but one gets more of the cytoplasm. The other one is a polar body. It's too small, it dies off. So they usually just die on their own. If they are fertilized, it's not going to create life. It's going to die because it doesn't have enough nutrients and cytoplasm and organelles to be a sustained life. So polar bodies are very short-lived cells that really can't do anything. So only one egg cell is produced in this process. While for sperm production, 4 sperm cells are produced during meiosis. So during meiosis, the egg gets most of the contents. The other 3 cells form polar bodies, which usually are very, very short-lived cells. So this process, sperm production is called spermatogenesis, and then egg production is called oogenesis. So oogenesis produces eggs, spermatogenesis produces sperm. They have similar processes where they're both meiosis, but the different kinds of cells produced at the end is what separates the two. Okay, so we're going to end today for notes. On page 166, work on questions 1 through 5. Turn the lights back on.